Thanks very much, Kathy, for a really fantastic talk, and really exciting work, and really the whole reason for me to have a job here. And what I'd like to talk to you about is something called ex situ heart perfusion, and the next steps in developing therapies for heart failure, like Kathy's special molecule, basically. And ex situ heart perfusion is a very fancy way of saying we look after hearts outside the body. Hearts are pretty simple things. They need some blood and they need some oxygen to work. And as long as you give them some, enough of that good stuff, they'll function just fine. This is just a little warning that the next slide is a pig heart being perfused outside the body, just as, just because some people might not want to see that. But if you do want to see that, this is what it does look like if I press play. So this is what a fairly healthy heart looks like. And you can see it pumping away. And the heart's amazing in that it, hopefully never stops pumping, because when it does, it's usually not a very pleasant outcome. And so this is a pig heart. We have done human hearts, which I'm not showing you here, but it's really the start of a project, a big project we're doing. And this is basically a brief outline of the machine we use. It's very simple. I know it seems like there's a lot going on. There's a pump and there's an oxygenator. So pump, ironically, to pump blood to a pump, that being the heart. So we have a pump for a pump. We have the oxygenator, which is just to give the heart oxygen. We have a water heater to make sure it's a fine temperature. And then we have a reservoir where some of the blood is stored. It's not too complicated. But if we just take a step back and look at how we make medicines and how we make therapeutics for people, well, it usually happens we find a molecule of interest, and that's a drug or an mRNA or a gene therapy or whatever it is. And, it seems promising. And then we do studies usually on animals, so rats, mice. And then when it looks a bit more promising, we move on to larger animals like sheep and pigs. We then go from those sheep and pig studies straight onto human studies in real life humans. And, that's usually okay. But it seems like we're making a big jump there. And then we go from those clinical studies, small scale clinical studies in a few people, to large clinical trials involving hundreds or thousands of people. And so we think there's a missing link there. We think it's a bit bizarre that we go from doing these studies on these animals straight to doing it in healthy or unhealthy sometimes living people. And there's no sort of in between. You're pretty much all in at that point. And animal models are really useful, but there's a very big difference between a healthy animal and a sick person. And that's why a lot of medicines, a lot of therapeutics fail at that stage of transitioning from preclinical to clinical studies. So we need models, basically, that can bridge that gap. And so we think by using ex situ heart perfusion, we can bridge that gap. They let us test a lot of therapies that we couldn't test in animals in humans. For example, if we want to test how well a human stem cell divides, we can't really test that in an animal because the animal's immune system will reject it because it's a human stem cell. But by doing it in a human heart, you could test that. A side benefit is that this could potentially reduce the number of animals needed for research, which is, as somebody who's unfortunately had to use a lot of animals for research, it's a benefit. It's something we're conscious of. We don't want to use animals if we don't have to use animals. So what have we done before? Well, we've used explanted hearts from transplant recipients, so people getting a new heart. We've taken their heart that's being removed, and instead of unfortunately just discarding it or putting it in a tissue bank like we usually would, we actually tested a new therapy that's designed to increase the amount of blood flow to an area. It's used to treat angina and heart attacks, which are both pretty prevalent conditions. Today in the UK, there will be 500 heart attacks, just today. So there's nothing special about today, by the way. That's every day. And so we perfused these hearts for 6 hours, and we showed that, whatever therapy it was, made a protein that would help increase the amount of blood flowing to the heart. And what's the impact of this? It was a great study, and it took us 10 months to effectively get 3 to 4 hearts. And really what we want to do is expand this. So in the UK, like we said before, there's 200 heart transplants a year. Of those, we think we could probably get about 80 to 100 of those hearts. The reason I'm saying only 80 to 100 is because about half of those patients will have these pumps in their chest, those ventricular assisted devices. That's a more complicated operation. When you remove a heart with a VAD, it's a bit more complicated, and we don't want to get in the way of a clinical program. So we just won't touch those. We'll say, remove the heart with the VAD, however you remove it, because our aim here is not to put any extra pressure on the surgeons. It's just to get things that are essentially going to be discarded. There are 800 hearts turned down a year for transplantation. Of these, we think we could probably use about half, just based off logistics. So it's about 500 hearts a year for research, which would make a massive difference. The number of therapy, you could easily test 100 new therapies for heart failure and other heart problems each year with that number alone. And to put that into perspective, in the last 16 years, this is data we've got from NHS Blood and Transplantation, only 35 hearts were used in research. That's out of a grand total of 6,300 hearts turned down. So it's less than 0.5% of hearts we're using for research. Or just over, actually, 0.5. There are some limitations with this. There's short perfusion times, so we can only look after hearts for about 6 hours. So a lot of my work goes into looking how we can long that out. How can we look after hearts for 12 hours or 24 hours? Currently, we have to apply for ethical approval for each of those studies individually, which is a very long process, and it's an important process. I'm not saying we don't need ethics to do this kind of work, but whether we could have a blanket ethics and just say, we want to test a series of molecules that we've done previous experiments with in animals on explanted, discarded human hearts. And there's a lack of infrastructure currently to use these organs for research. And really what our long-term goal is to have a national organ retrieval service for discarded organs. So for turned down donor hearts and for explanted recipient hearts, which otherwise wouldn't be used for research. So our ultimate aim is to improve our tools of drug discovery. I'm not here going to cure heart failure, but what I want to do is help people like Kathy who are probably going to do, at least make an impact on heart failure, so that we don't need to, keep having as many transplants. And because transplantation is fantastic, but there are people who don't get transplants. There are people who die, unfortunately, on the waiting list, people who are removed on the waiting list because they become too sick. So I'm delighted. I'm delighted you've all gotten transplants. I think it's a fantastic operation, but like we've, I think, hopefully established, it doesn't work for the overwhelming majority of people with diseased particular organ.